So the next speaker is Professor Paul Matthews, who's going to talk about repeat imaging in Biobank. Thank you, Paul.
Thank you, Julie. It's wonderful to be here again this year. Each year that I come to these Biobank Scientific Conferences, the science becomes more exciting, the opportunities in UK Biobank become greater, and the richness of the data store rises. I'm going to talk today about one of the most important aspects of UK Biobank is not only the richness of the data, which I will touch on in terms of making precision analyses of the different forms of dementia, but also in the fact that it is prospective and longitudinal. Now, as the extraordinary opportunity for those of us interested in dementia that is afforded by UK Biobank is that we now have an opportunity to watch the development of dementia as well as other diseases of late life before they happen. We can see those factors which are important, unconfounded by reverse causation. If you look at the data for the estimated incident disease numbers for Alzheimer's disease, by 2027, it's almost 20,000. By 2032, it's 40,000. These people between this number and these numbers are now within the cohort, and we have an opportunity to watch them, to learn from them, and to help develop the kinds of treatments that will reduce the risk for others and prevent disease in some. One of the, amongst the several enhancements that have been made in the UK Biobank resource over the years was the imaging enhancement, and some of the benefits of that were referred to in David's talk. But what I want to talk about is, despite the fact that I'm a neurologist and fundamentally interested in this organ, I have relearned my medicine and have become interested in all of these organs. And the extraordinary thing emphasized by David in his talk about multimorbidity is that diseases of the brain in late life do not occur in isolation, but occur in the context of diseases of the body. And the ability to link brain imaging, precision phenotyping with those for heart, body, lean mass, fat mass, liver, looking at large vessels such as with carotid ultrasound, bone with DEXA to understand frailty in a more granular way, and also to see the volunteers within UK Biobank again, take more measurements in person, and with their generous contribution of additional biofluids, develop material to allow serial observations of markers in the blood, some of which we'll hear about later this year. And of course, from all of this, we can derive new kinds of measures that are more precise in their descriptions of pathology. Now, this is all very exciting, but this year a major advance has occurred with the generous funding from the Chan Zuckerberg Initiative, Calico, and the UKRI Medical Research Council to UK Biobank to enable the re-imaging of over 60,000 of the participants beginning this year and continuing through about 2028. The extraordinary task of imaging almost 100,000 of the participants has been made even more exceptional in its opportunities by allowing serial observations with time intervals of 2 to 7 years between imaging visits for about 60,000 of them, as is estimated now. Now, in addition to this, I'm going to describe briefly not only will the re-imaging occur, but UK Biobank has worked with the scientific community and the funders to allow additional enhancements of the imaging visit, including retinal optical coherence tomography, and I'll show you an example of that later. Enhancements in the brain imaging protocol itself. We've been particularly aided by Steve Smith and his group in Oxford, who have helped develop an arterial spin labeling protocol for cerebral blood flow measurements that is now being added, and of course working with a variety of stakeholders to enhance the cognitive measures in the data in the resource. Now, even as a neurologist, I used to find neuro-ophthalmology the most difficult aspect of my field. But even as a neurologist, I've come to get very excited about retinal optical coherence tomography, because it is the only way that we can, without removing the skull, look at brain tissue, neural tissue directly, and its relationship with the arterioles that supply it. We look in the back of the eye using a laser scanning device, and within a minute, one can develop a very detailed measure of the thickness of the retinal layer, which is a measure of the density of neurites in that area, as well as looking at a variety of blood flow measures. We've only just started with the ophthalmology community to mine these data from the initial set, but now we're going to have an opportunity to acquire new data and to link this directly with brain images acquired at the same time. Now, again, part of the enhancement of that will occur in the re-imaging visit is what for current thinking about the onset of dementia may be one of the most important additions, and that is a measure of cerebral blood flow. Investigations thus far using ADNI, while incomplete still, have suggested very powerfully that changes in blood flow may be one of the earliest elements of pathology in Alzheimer's disease before it develops, or at least in the early stages, and may be a driver of other pathologies. Now we have an opportunity to test this prospectively and to define it much more precisely with work by Tom Okel, Michael Chappell, and other colleagues in Oxford. The initial pilot data, remember, everything that goes into UK Biobank is piloted extensively before it ever really becomes part of the protocol to ensure that the quality is sufficient to allow all of us to use it. Here they've shown in the initial data set that, for example, these measures of cerebral blood flow, here you see a map of cerebral blood flow in mils per 100 gram per minute with lighter colors indicating higher blood flow. You can see the clustering of higher blood flow around the edges where the neural tissue predominantly is found, and in the center. This is complemented by a measure of arterial transit time, which, crudely speaking, is the rate of movement of blood through the vessels, a complement to a measure of cerebral blood flow. You can see these maps. You can look at the age-related changes over the global cerebral blood flow and see how this varies in this population with age. This is 85, this is 50. One can also look at the arterial transit time to get a similar measure. In some ways, it can be a little bit more informative. This is terrifically exciting because we can test the relationship between cerebral blood flow changes, brain atrophy, changes in the microstructure that are associated with it, and cognition. Now, of course, the re-imaging itself, the repeat of precisely the same imaging measures at the second time point as those at the first time point is fundamental to understanding the longitudinal course of any disease or pre-disease state. And this really just illustrates in a very small way from another study the importance here. One has looked at a variety of snapshots of serial measures. This is years. One measure was acquired at one point on the curve, another at one or more points subsequent to that. And you can see that individuals in the population, illustrated here as these red lines or these blue lines, individuals in the population show widely varied trajectories of organ-specific longitudinal brain change, which can only be detected if you do serial measurements as opposed to, imagine, just taking a cross-sectional measure. These serial measures, by looking at the slope of that line, one can much more powerfully distinguish disease groups, shown here in red in the case of dementia, versus those who are not developing disease within the period of observation, those in blue. This will now be part of UK Biobank in a big, big way. I'll give you one example that's already come, which is just an extraordinary example, again, from the Oxford group, Steve Smith, Carla Miller, Gwen Dou, who led a team looking at the effects of SARS-CoV-2 exposure during the first waves of the pandemic on brain changes and on cognition, using the pilot for the UK Biobank re-imaging protocol, which was a small number, about 3,000. It was set up by the Biobank team incredibly quickly as a nested case control study, a unique thing within UK Biobank. And what Gwen and the team were able to study is the change here in brain volume measures with time, pre and post-pandemic, distinguishing between those people who had no serological evidence of exposure to the SARS-CoV-2 virus and those who did. And what is extraordinary, indeed disconcerting, is there were significant differences when corrected for the appropriate demographic variables, and these could be mapped here, shown in blue. This is a brain, a blown up half of a brain, flipped on its end here in the inferior cortex. Now, much more is going to be happening. When I come back in future years, there'll be much more talk about algorithms for defining specific clinical diagnoses, expansion of the imaging protocol, new kinds of devices, such as digital devices, more omics measures in blood and others. And we'll be able to really begin over the next 5 years and more to answer key questions. What are the genetic risks for different types of dementia? How do environmental and lifestyle factors contribute to impact on this genetic risk? How do they vary between ethnic groups? We mustn't forget that they are represented within UK Biobank. And then how, most important for treatment, how can we recognize dementia in its preclinical phases when we want to treat it? So I want to just close by thanking everyone in UK Biobank who's made this possible. It's a huge team effort. They've done enormous work, the funders, and of course, the 500,000 participants across the program. Thank you.